Okay, it has DNA or it has nucleic acids. May not have DNA, but
Does it respond to stimuli?
It has a very basic intelligence.
okay, it responds to stimuli.
That's a better way of explaining what I just said.
Yeah, intelligence is a sticky word.
Yeah.
So it can, it won't really like go towards light, but it has specific receptors that it can attach to. Right? Yes. It does change over time, I guess. Or it will evolve. Okay, and that's actually really important for us if viruses are evolving and changing. Okay, anything else?
I have a question.
Yes.
Okay, so when you talk about like viruses changing and evolving, so then technically they have like an ancestral tree, right? They have ancestors, so
Yeah.
you could classify that as like having a 5% tree, right?
Okay, yeah, so they do have ancestors, and you could trace them to a common ancestor in a phylogeny. That's true. Yeah, this is a byproduct of it being able to multiply and reproduce. Yes?
This is kind of an abstract theoretical question, but if we're going to count viruses as alive, then couldn't we count computer programs as alive, self-replicating computer programs?
Well, I guess. I don't know.
There are neural networks that learn
You
and
can-
reproduce themselves.
Yeah, can they do it continuously
As far as
without a power source?
without power
No, probably not. So I don't know. I mean, what we are actually listing are characteristics that it shares with things that are living, but there are a lot that it doesn't share, right? And so yeah, there are some characteristics in like computer programs which share some characteristics, but probably even less than viruses. But good question. All right. Why would you qualify it as not alive? What does it not have? Yeah.
It can't survive or replicate without a host.
Okay. It needs a host, so it's not independent. Okay. What else? Oh, so there's another one we should put over here, right? They have proteins as well. Okay, and they have nucleic acids. Okay, but they don't use carbohydrates and they don't use lipids. Why not?
Do they not use energy?
Yeah, they don't use energy, or they use energy. They use ATP, but they don't produce it, right? Yeah, so they can't metabolize their own, they can't make their own energy, right? And that's part of needing a host, not being independent. They're also dependent on the host energy that's available.
So with energy then, does that mean that viruses can't like regulate their temperature and
Right. Yeah, they can't really do anything with homeostasis. They are at the mercy of their host. Yeah. Okay, what else? Okay, they have proteins and nucleic acids, but what do they not have? We said lipids and anything else? All right, what else defines life? Yes? Right? They do not have cytoplasm, which is where you would find those energy molecules. What else do they not have? Arms, good. Well, some of them might. Yeah.
They don't have cells.
There you go. That's what I was looking for. right? They do not have cells. And if we are going to find the fundamental unit of life as a cell, then they wouldn't fit. Yes?
So going back to not metabolizing, how do they do things if they're not making energy for themselves? Do they just start with a set amount of energy and then just die?
No, they don't have any energy. They're just a structure floating around. When they go inside a cell, they use whatever's in the cell to replicate.
Okay.
And then they burst out of that cell. But by themselves, they can't do anything. They have to find a cell to invade. Yeah.
So they just float, no have like direction.
Yeah. All right, they don't have a flagella, so that's another thing they can't do. They can't move independently. There's a lot of little living things that can't do that as well. Okay? But I didn't spell independent very well. Okay? But yeah, they are simply at the mercy of diffusion and osmosis and just floating around until they find something. Okay? But there are a lot more viruses than there are organisms. A lot more different types, but each of them has to be specific to a living thing in order to survive. Okay? But this is a good list. Anything else you would add to this? Okay. All right, so stop doing that. Which came first then, bacteria or virus? Discuss with your neighbor.
Oh, virus needs it though, so it can become more bacteria, which are like the food or something. Yeah, I think the virus can
All right, I think this one was easier to comprehend. So which came first? What? The chicken or the egg? It's obviously the egg. There were a lot of things. Dinosaurs laid eggs, and chickens are birds, which came from dinosaurs. So the egg came first. Well, it's not dinosaur and egg, it's chicken and egg. Anyway, all right. Which came first, bacteria or the virus? Bacteria. Why? Virus needs a host. The virus is not independent, so there's no way it could do anything without itself. Yes?
But what if they were both created in Precambrian explosion, and the virus only started to replicate when it first came in contact with the bacteria?
They develop simultaneously.
Wouldn't the same molecules needed for both of those organisms, both of those things, be present?
Except that bacteria needs more,
Yes.
right?
So
And
how
a
do you-
virus needs a bacteria.
to replicate.
Yeah.
But if we have all of the molecules necessary.
So it could have been floating around without a host.
Yeah.
Sure.
So technically,
Sure, but it's more likely that actually a bacteria existed with DNA and enzymes and everything to replicate, and then something went wrong and a virus was created. Yeah. So or a cell maybe lost there's actually a bunch of different theories about this, but one of them is that a cell lost some of these other features which made it a cell, and then what was left was a virus. But yeah, that one is hard to determine because that's not. But viruses don't fossilize, right?
so then viruses wouldn't have come much after the bacteria.
They must have come after bacteria, yeah, but maybe not much.
Okay.
Yeah. Good question. And there's a host of literature out there discussing that if you want to get into it, but I don't know much about it. Okay. All right, so what are viruses? They are, they have a few simple things, which we stated in the other live and not live, but they have a nucleic acid. Okay, what are our different types of nucleic acids? RNA and or DNA. They usually have one or the other, don't have both. Okay, and then they have a capsid. Okay, what's a capsid made of? Protein, good. Okay, and they may also, we're going to put 3 on here in a different color because not all of viruses have this, but they might also have an envelope surrounding their capsid and their nucleic acid. But not all of them will have that. Okay, within that viruses, and they may also have enzymes. Okay, they may also have some enzymes which help them hijack their host machinery. Yes?
So I didn't quite understand the first question on the quiz. The one that was like, are there viruses? No, it was viruses only have RNA or something. I don't know. It kind of seemed to ask if there are viruses that contain more than just RNA.
All right, we'll have to look at it later.
Okay.
But yeah, I don't know.
I got right.
I don't know.
Okay. But yeah, viruses at least have nucleic acid and capsid. They may also have envelopes and enzymes. Okay? There are different ways we can classify them, either by shape. Okay? So some of them have a... A capsid that is icosahedral, which means it has many sides. All right. What were some of the other shapes? Bacterial-like? Oh, the bacteriophage. Yes. Right. So the bacteriophage is has 2 parts, has a head and a tail. Okay. It's binal is what it's called. That just means it has a head and a tail. Okay, what else? It may also just have a spherical capsid or a rod-shaped capsid. Okay, but they're generally very simple in structure. Okay, the most complicated it gets is the bacteriophage, which has like a head and a tail with little arms. Okay? Beyond just having DNA or RNA, they also might be single-stranded. Oh, I've got that down here. Or double-stranded. Okay, and you can have any combination of that. Single-stranded DNA, double-stranded RNA, single-stranded RNA. Okay? And they're also classified by their host. So, for example, H1N1, what was that? Does anyone recognize that name? The swine flu, okay. So this is a virus that actually has mutated through multiple hosts, of which you have, of course, pig, chicken, and human. but most of it was from the pig. It's called the swine flu. But this is a type of influenza virus, and influenza has those multiple hosts, humans, chickens, and pigs. Okay, so in areas where humans live in close proximity to their things that they eat, such as pigs and chickens, they're more likely to develop these new strains of flu, which can then spread throughout the world, which happened with the H1N1. And there was a big scare. Did anyone get the H1N1 vaccine?
I got H1N1.
You got it. Oh, okay. Well, then you got the vaccine, right? Naturally. But yeah, if it's a new strain which you don't have the immunity for, they can get a lot of people sick really fast. And not very many people died as many as they thought would, partially because the awareness and the vaccine that was developed for it.
There's so many, there's some viruses that can rapidly mutate because because of the environment. Like, for example, bubonic plague, when they still live with the chickens and the pigs inside the house. Is that why it's able to mutate so rapidly?
Yeah, so the hosts were very close to the exposure to each other constantly. And it just takes one variant that can jump hosts to now make a very new virus that you don't have an immunity for or any sort of similarity. So the same thing happens with Ebola. So Ebola came from bats, is where they think it comes from in Africa. And there are different strains that have jumped from bats to humans, and then it's very deadly in humans and passes very quickly. Did anyone get Ebola here? Hopefully not. Yeah. Twice. So Ebola has a very, very high death rate. It was somewhere near 50%, which half of the people who got Ebola survived, but half of them died. That's quite high, right? So your influenza flu virus that passes through is much, much less than that. Yeah, lower than 1%. Okay. So one of the enzymes which is really important for many of the viruses is called reverse transcriptase. Okay, and this is an enzyme that works backward. Okay, so we're going to actually have you think a little bit back to first semester. What is the central dogma? See if you can remember between you and your friend. Yeah, so I just corrected that RNA. There was a T in there, and it should have been, right? All right, so what's the central dogma? DNA to RNA to protein. All right, what's in between this DNA and RNA? What does that arrow represent? Okay, transcription here. And from RNA to protein? Translation. All right, so reverse transcriptase is used when, we have natural enzymes that use reverse transcriptase, but for viruses, it is most commonly used when the virus has an RNA molecule. And what it does is it goes from RNA to DNA. Right? So if this was the RNA of this virus, what would the DNA be if it used reverse transcriptase? Remember this from last? What's that?
Is it kind of translating itself into the cell, writing itself into the cell?
Yeah, that's what it does. So
That's so cool.
It uses RNA as a guide, and then reverse transcriptase makes DNA from the RNA. So then this would be well, I'm all messed up. TCGAT. Okay. All right, so that's the DNA it would make. And then it would make another strand of DNA. And now it has a double-stranded DNA, and then that DNA would insert into the genome of the host. And then it would go and make the RNA again, a different RNA, which would then be transcribed or translated into proteins, which would make up the viruses.
And this is what bacteria does?
This is what the virus does with reverse transcriptase within the bacteria or the host or whatever it is. Yeah.
So, like last semester when we learned about like the Cas9 and all that, they take it from the bacteria, right, or the virus. They found that in them. So is this what it's doing? It's
The RNA guide, what are you saying?
well, you know how they like found it in the bacteria, the
Right.
mechanism. So is this the mechanism that they found, or
No, it's something separate. Yeah, it may use reverse transcriptase, but I'm not sure at which step.
okay?
Yeah.
So which one of those DNA things are being inserted into the genome?
So both of them, because DNA is double-stranded, so it needs both strands to go into the genome, and then once it's in there, then it can then be transcribed and translated. So if an RNA virus is essentially adding a step to the central dogma. So it's going from RNA to DNA to RNA to protein. And it's using reverse transcriptase to do that. But reverse transcriptase and viruses are used in biotechnology or genetics to, if you can make an RNA which codes for a specific gene, you can put that RNA into a virus with its reverse transcriptase, and then take that virus and infect a cell, and then make whatever product you want. Okay, so it's used in a lot of biotechnology practices for gene editing and inserting genes and things like that. Yeah.
Is that how they front insulin?
Yeah. So they used reverse transcriptase in that as well. Okay, yeah, and I think we mentioned it last year when we talked about biotechnology. Yes?
In that case, when we try to remove the virus from the body, are we essentially telling our body, we're putting something in us that is attacking our own cells?
Yes, so most of the ways in which your body attacks viral infection is by finding the cells that are infected and killing those cells and removing them. Okay, and then you kill enough of those cells and then you get rid of the virus. Viral infection. Yeah, yeah. So viral infections can be really bad because they've already infected a lot of your cells. Whereas a bacterial infection, if you just kill the bacteria, now you have to kill your own cells.
So in a way, it's kind of similar to killing cancer, because you have to attack your own cells.
Yeah, similar way. And your immune system will sometimes detect cancer cells, put up this flag, and another immune cell will come and kill that cell. So similar process. Yes. Okay, any other questions? Okay, so then the way that this all comes together has 4 steps for how a virus enters into a cell. Okay, the first step is attachment. Okay, so within the cell has, specific chemicals within it, and different cells have specific types of chemicals. And the virus, I'm going to draw kind of a bacteriophage. But it recognizes then these specific signatures of the cells. Okay? And it generally only has a small number of, or specific types of cells that it can infect. Okay? So it has to recognize and then attach to that cell first. The second thing it does then is it goes into the cell. Okay? And what are different ways that you think thinking of how things move through plasma membranes, what are different ways you think that this bacteria or this virus could move through? Yeah.
Phagocytosis.
Okay, good. Yeah, endocytosis is one way where it takes part of this membrane and then is within that membrane. Okay, another way is it just may find pores that it just fits in between and recognizes that it can go in there.
Is it possible that they have the same kind of proteins that are surrounding it as other cells, and so that they just go through because of the
It just,
membrane?
yeah. So you're saying their protein is similar enough to where,
It allows it through the membrane?
yeah. So it could go through specific channels or pores through the membrane. Yeah. Once it's in there, so within our little cell here is our nucleic acid. And it has the enzymes in there. But then it has to replicate. So we described one of the ways using reverse transcriptase that it could then go into the nucleus here. And then it's going to use some of the enzymes which are already in the cell and or some DNA, all right, double-stranded DNA. And then it's going to replicate the mRNA and go through the process of transcription and translation. All right, the last thing that it's going to do then, once it has all its pieces, is it'll make a bunch of these new little viruses. And then exit the cell. Okay? As it is floating, wherever it is, and before it attaches to anything, it is inert, meaning it can't really do anything, right? So it is called a virion. Once it has found its host and it's replicating, it's active. In this form, it is a virus. Okay, so as an infecting agent that's taking over machinery of the cell, it's a virus. As it's floating around in your saliva or fluid or air or wherever it is, it's a virion. Yes?
So is it only a virus once it gets into one of the organelles, or just if it gets into the
Once it gets into a cell, yeah, then it's a virus. It is now in its infectious stage. Yes?
When it attaches, though, it's still a
I mean, you could say that within there, it's starting its infection at attachment. Yes. Yes.
So with entry, when you said there's like pores, is that why we see like some have the envelope or?
Mm-hmm. Yeah. So sometimes the envelope actually has the recognition of the cell on the envelope, and then, well, I'll show you a video actually in a second. So it'll have different ways of entry in there. Okay. Any other questions? And it's, whoops.